So this first question was from 2024, so it's from last year's paper, and there were two parts to it, right? So the first part, actually both these questions talk about the idea of crossing over and independent assortment. I'm just going to write them on the board because we're going to talk about them for both questions. Can someone tell me what crossing over is?
Chromatids exchanging and alleles exchanging within the chromatids.
Okay, so chromatids crossing over, right, and exchanging alleles, right? And when does this happen?
Prophase.
Yeah, prophase 1. And can someone remind me what's an allele? What's an allele? Yes, Paramveer? Yeah, right. It's a variation of a gene that codes for a specific trait, right? So generally, maternal paternal alleles will be a bit different, right? So that's one method of variation in meiosis. The next one we have is independent assortment. Right. When does this happen in meiosis? Can someone tell me? Yes.
Metaphase?
Yeah, metaphase what?
1.
Metaphase 1. Right, and can someone, actually I'll just explain, right? It's just how the pairs line up on either side of the plane, right? So you can either have your pairs lining up like this, right? Or maybe they decide to line up a bit differently, like this, and then they get pulled apart in anaphase, right? And that gives you a different end product cell. Yes, Arya?
Are these both things that happen, or is it like either or?
They both happen.
Okay.
Yeah, one after the other. Yeah?
For the crossing over, it says on the notes that it is between prophase 1 and metaphase 1.
Yeah, that's true. That's because some people kind of refer to it as prometaphase. But just for simplicity, we'll just say it occurs in prophase 1.
Okay.
But I'll show you guys the marking criteria for the question. But actually, the HSC didn't require you to know exactly what part of meiosis it happened in to get full marks. It just said meiosis 1, right? So as long as you know that both these things occur in meiosis 1, for the particular questions you did, that was probably actually enough, which is pretty good, right? We don't have to remember it. So why is crossing over so good for these little jack jumper ants? Can someone tell me? Yes, Arya?
Genetic variation.
Right, cool. Crossing over gives us genetic variation, right? It's a 2-marker, so it's probably it's an outline question. So there's not too involved, right? So let's just say increases genetic variation. Right now, if you read the question carefully, it says outline the significance of crossing over, right? So genetic variation happens. I think for this question, we actually have to also say why genetic variation is significant, right? So just a general statement on that will do. Can someone tell me why genetic variation might be important? Thinking back to year 11. Yes, Hannah? Yeah. So variation in the species is good because you can adapt better to different environments with different traits, right? So I might just say increased survival chance due to variation in traits. All right, now this next question, B, I saw us struggling with a lot. So I'm going to walk us through it, right? So draw the chromosomes of the 4 possible gametes after crossing over for the jack jumper ants occurs. Include the alleles for each gene. Now, what I'll do is you have to do this correctly. I saw all of us drawing chromosome pairs. To do this correctly, you actually have to read the question very carefully, right? Female jack jumper ants have a single pair of chromosomes. During meiosis, crossing over occurs, right? So I'll show this on the board, right? Actually I'll show this here. My jack jumper ants, right, they only have a single pair of chromosomes, right? So before interphase happens, they have a chromosome from their mum, right, the maternal one, and one from their dad, the paternal one, right? That's their entire, like, that's it, right? Then after interphase, we know that the maternal one will duplicate. So then I'll have 2, and the paternal one will duplicate as well, right? And that means my cell, after interphase, after the DNA contents have been replicated, will look like that, right? The second one. And that's what I see in this diagram here, right? My homologous chromosome tetrad with various alleles on it, right? Now, in meiosis, right, we know that there's 2 sets of division in meiosis 1 and meiosis 2. So I'll just show you what should happen in each stage of meiosis and what the chromosomes will look like without considering crossing over first, right? So this is my cell, right, before meiosis, when the cell contents have duplicated. Then, right, it splits, which I'll have to move. Right, then it splits, which I'll draw here actually. Right, so I have my one cell, right, and then it splits into 2, right? I'm not going to worry about the processes for genetic variation just yet. I just want to show you how the numbers will look, right? Right, so this is meiosis 1, right? The first stage is done, the first split. But then, you know, meiosis 2 happens and the split happens again, right? And I'm halving the genetic material every single time, which means that my resultant 4 gametes will just have a singular chromosome. 1 chromosome, 1 chromatid, right? Can we see that? Yeah? Yeah, so I know a lot of us are drawing pairs because we expected there to be pairs based off like various diagrams, but no, it's just 1 chromosome, 1 chromatid, right? So all we have to do is draw the 4 possible combinations just singularly, right? So just looking back at this diagram here, what are my possible combinations? Well, I have my combination before any crossing over happens, right, which is this one. So combination 1 is big D, big E, and then big H, right? Combination 2 is just when nothing else happens. So this one. So lowercase d, lowercase e, lowercase h, right? My third combination and fourth combination is when crossing over has happened. And we can see in the diagram, crossing over happens at the bottom here, right? Where these 2 genes are. So my 2 crossed over, right, would either be capital D, capital E, and lowercase h, right? So this one here. Or it would be lowercase d, lowercase e, capital H, right? So I just need to draw these 4 gametes on the bottom here, which I will get for us. Right. Cool. So right, lowercase d, lowercase e, lowercase h. I'm just going to signify using colors. I don't think you should use colors in the HSC. I'm pretty sure they tell you not to, but you know, for the purpose of, since I can, I will, right? And then lowercase h. Lowercase d. Yes? Yeah, it can just be a line. It can just be a line.
Do we have to say like the D?
Yes, yes, you should, because you're specifying the alleles that are present. Oh, crap. Okay. All right. Our 4 gametes should look a bit like this.